Do you remember biology classes in school? One of the first things they teach us is that the DNA is like the instruction manual of every living being. It is made by the repetition millions and millions of times of four letters A, C, G, T, called nucleotides. Then, after the cell makes a copy of it, the DNA in the form of chromosomes is divided equally between the new cells. You may have seen a chromosome somewhere before, and they're usually depicted with an X shape. Maybe the one you saw looked like this. The narrowest part of the chromosome is called the centromere, and this is the structure that is essential for the correct and equal distribution of the DNA between the new cells after each cell division. Another thing we are taught early on in those classes is that in the case of the DNA, its sequence is extremely important. In fact, unfortunately, some diseases are due to changes in the nucleotide sequence. So now you may be wondering whether this is the case for the centromere as well, and if the centromere is also specified by the presence of a specific DNA sequence. But the answer is no. The centromere at the DNA sequence level consists of stretches of these nucleotides that are repeated thousands of times. However, some chromosomes in some animals, like horses for example, lack this repeated sequence at their centromere. But they're still among us and they're doing just fine. Then the centromere must be defined by something else, right? Yes, this is the case, but before we go there, we need to take a step back. Do you remember at the very beginning when I said that the DNA is like the instruction manual of each of our cells? But now look at us, humans for example. We are so complex. Then the DNA molecule must be so long, right? Yes, in our cells, the total length of the DNA in one cell is 2 meters. Isn't it amazing? And now another question arises. How does it fit into the cell? Cells are so small we can't even see them without a microscope, right? Yes, so to fit into the cell, the DNA is compacted in a structure that resembles a pearl necklace. Now imagine that each of the pearls of this necklace are made of proteins that we call histones. At the centromere, these pearls are a bit different because one of the histones is replaced by another histone that is called SNP, centromere protein A. So now you may have guessed where this is going. This SNP protein is only present at the centromere and nowhere else on the chromosome arms. So this is how the centromere is specified by the presence of this protein SNP and why it's special.